A very good morning to all. To begin with, I would like to thank the organizer for allowing me to present my work in this conference. Today, I'm going to talk about arsenic resistant bacteria, which is a future weapon to combat arsenic toxicity in soil and groundwater. I also thank my co-authors, Ms. Debjani Mondol and Mr. Ritesh Shonar, who also belong to the Department of Molecular Biology and Biotechnology, Sripat Singh College, affiliated to University of Kalyan. So our college that is located in the state of West Bengal, this is India. So we have the state of West Bengal near this Bay of Bengal, where this JBM river system is active. Ganga Brahmaputra Meghna river system is active, and that is one of the primary reasons for arsenic contamination of the soil of this Gangetic deltaic region. In the state of West Bengal, there is a district, Murshidabad. This district is a extremely backward district. Our college is located in this district, and we have done our work in this Bhagavan Gola block of Murshidabad district, okay, which is extremely arsenic contaminated. So this is the Murshidabad district and Bhagavan Gola block. I'll show you another image of that. And this is the region, this Asanpada region, okay, where we have mostly done our work regarding the isolation of arsenic resistant bacteria, as well as studying the clinical manifestation of arsenic within the socio-economically backward villages of this Asanpada village. So here we can see that within this Murshidabad district, these are the 9 blocks which suffer from tremendous arsenic toxicity. Now we are going to talk about the arsenic toxicity or the arsenic concentration present in the groundwater samples that are basically extracted by tube oil in Bhagavangola 1. So see, this is Bhagavangola 1 block. As you can see that most of the water samples contain more than the recommended or the maximum permissible limit of arsenic that can be present in the drinking water samples. Here I would like to make a point that most of the drinking water used by these villagers comes from these tube oils, which are the sources of groundwater. Okay, so if I go to our next slide, we know that US EPA they prescribe 10 microliter, 10 ppm, 10 ppm as the highest permissible limit of arsenic that can be present in the drinking water. WHO also corroborated, but they said that in absence of alternate source, you can have 50 ppm of arsenic in drinking water. If we just see the state of this Bhagavangola 1 block, we'll see something which is very alarming. That is, almost 61.2% of the groundwater samples extracted from the tube oil, they contain more than 10 ppm of arsenic. 30.6% of the samples even contain more than 50 ppm of arsenic. And the maximum arsenic concentration in this Bhagavangola 1 block of Murshidabad district is almost 1.3 mg per liter. That is almost 1285 ppm. Okay, even you can see there are other blocks which are even more contaminated, which have 2000 ppm of arsenic, or we have almost 80% of the samples, groundwater samples, which contain non-permissible limit of arsenic. Now, some of the clinical manifestation of this arsenic toxicity that we have seen in the villages of Asanpada village. Okay, and this is due to the prolonged exposure to arsenic, be it in the soil. So when arsenic is present in the soil, it is getting biomagnified within the rice cultivars, and when this rice is taken, it gets within our body, or the water samples are contaminated, whether for washing, drinking, or any other kind of household chores, they are consuming arsenic contaminated water. We could see some preliminary observations here pertaining to arsenicosis with multiple nodular growths, keratosis, hyperkeratosis, melanosis. Okay, then hyperpigmentosis, darkening of nails. Then there are disorders of different physiological systems, even going to the extent of cancer of liver or urinary tract, and death in extreme cases. So the extreme cases of arsenic toxicity can only be surveyed, but here we have presented some clinical manifestations. These are our preliminary observations. Now, from the soil of Bhagavangola 1 block, to be more precise, from the Asanpada village, we have extracted two, or we have rather isolated two arsenic hypertolerant bacteria. Okay, by plating this bacteria under arsenic stress. And what we could see is that in terms of being tolerant to arsenic stress, these bacteria are completely, they are unmatchable. They show unprecedented tolerance to arsenic stress. We have these bacteria showing almost 77 millimolar and 88 millimolar concentration of arsenic they can bear. Arsenite, that is the most toxic state. Okay, plus 2 is the most toxic state. And 560 millimolar and 720 millimolar respectively of arsenate concentration they can bear. Also, they show hyperresistance to other metals like cobalt, chromium, and copper. Now, somebody may be curious that how we came to know regarding the name of this bacteria. I'll tell. So initially, what we have done is we have presented a scanning electron microscope image of this bacteria. Here we can see the scanning electron microscope image of the bacteria. Then we did the characterization of this bacteria. Some of the important characters which we are planning to exploit in our future research is the ability to break urea, that is the urease, as well as its ability of triglyceride degradation. Now, these have roles in agriculture as well as in bioremediation. But that's secondary. In this lecture, we will talk about the arsenic bioremediation ability of this bacteria. So, by standard 16S rDNA sequencing, we have constructed two phylogenetic trees, and we have identified these two bacteria as Lysodibacillus sp and Bacillus capensis. Okay, so by 16S rDNA sequences, we have identified these two strains of isolated arsenic resistant bacteria. Now, when we have grown this bacteria at extreme arsenic stress, like in 1000 ppm of arsenic, okay, what we could see, the bacteria is showing, or the bacteria are showing the same growth pattern in the control culture as well as in 1000 ppm of arsenic. Strangely, in 2000 ppm, their growth gets reduced, but once we grow the bacteria in 1000 ppm and then we transfer it into the 2000 ppm containing media, again we show the growth rate gets increased. So we are extremely curious that how can a bacteria show similar growth rate in control medium, that is the medium containing no arsenic, and in presence of 1000 ppm of arsenic. Is it like that this arsenic is helping in the growth of this bacteria, so that the heavy metal stress encountered by this bacteria gets negated due to the utilization of arsenic as a source of energy? Is it some kind of an assimilatory process occurring within the bacteria? That is, the bacteria is assimilating arsenic from the medium into the cell, or is it kind of a dissimilatory process? That is, this arsenic, that is, we have this concentration is mainly of arsenite, that is the +3 state, which is the most toxic state because it inhibits different enzymes essential for biochemical activity. Now, can this be transformed into a less toxic state like the +5 state? Is this dissimilatory mechanism of biotransformation that is occurring within the bacteria? But we are sure that something is occurring, that even at this high arsenic stress, the bacteria is showing a significantly good growth pattern. To check that, what we have done a biotransformation experiment. That is, we have grown this bacteria in arsenite, in 500 ppm of arsenite. Now, within the bacterial plate, we have added silver nitrate. If there is a formation of brown colonies, that means it is converting the arsenite into the arsenate. And we could actually observe that these bacteria are capable of converting the arsenite into arsenate. So this is an example of biotransformation. Now, we have two bacteria which show unprecedented growth in arsenic containing medium, unprecedented tolerance to arsenic, as well as some other heavy metals like cobalt, copper, and chromium +3. How can we use this bacteria? How can we use this bacteria to solve the problem or the arsenic menace faced by the residents of Murshidabad district? There begin our next cycle of work, where we have used this bacteria for the purpose of bioremediation. What we have done by SGDC assay, we have measured the arsenic concentration, the arsenic-free or the arsenite concentration. We could see that in presence of this bacteria and in presence of 5 ppm of sodium arsenite in their media, the arsenite concentration gets reduced by 32% approximately for Lysine-Bacillus and 37 approximately for Bacillus saphensis. This is for 5 ppm of arsenite. It could reduce the arsenic concentration by 32 or 37%. Now, if we increase the arsenic concentration to 1000 ppm, which is a very high concentration of arsenic, but still we want to see if our bacteria is capable of showing its ability of bioremediation at such high concentration of arsenic, because it's capable of growing at that high concentration of arsenic. And surprisingly, we could see that Lysine-Bacillus could still remove around 22.5% of arsenic at 1000 ppm arsenite concentration and 27.7 ppm of arsenic at 1000 ppm arsenite concentration is removed by Bacillus saphensis. Saphensis could remove 27.7%. So, what we can conclude is we don't know the mechanism. Maybe there is biotransformation of arsenite to arsenate, or there could be accumulation of this arsenite within the bacterial cell. Either way, it's removing a substantial amount of arsenite from the culture medium. This is very promising, and our future course of work is guided by our two projects, where we have said that we are going to use this bacteria for bioaccumulation of arsenic from soil, groundwater, and wastewater. So this is a treatment we are going to give to the soil, groundwater, and wastewater to substantially mitigate the arsenic toxicity in this soil samples, groundwater, and wastewater samples, as well as we are interested in designing a filter where there will be a bioremediation technology, and it will be supplemented by some chemical methods where we will use a class of novel eco-friendly surface modified metal oxides, and these two methods will complement each other or rather supplement each other for removal of arsenic from the groundwater sample. And we propose to provide a portable household filter for this arsenic infested water treatment. So thank you very much. And if there is any question, I'll be very happy to